and very welcome all of you, both you who are here with us in the Lund office of RISE, and also all of you online participating. So my name is Alexis Pirinen, and I'm a machine learning researcher at RISE. RISE is, let's see here.
I'll take care of it.
Okay. So RISE is Sweden's research institute. We have a broad variety of topics, including AI, machine learning, and cybersecurity. This is our weekly seminar series on the topic of AI and machine learning. And this meeting will be recorded. If somebody wants to be removed, please let us know. And today I have the pleasure of introducing Jakob Ambsdorf, who is a PhD student at University of Copenhagen and the Pioneer Center for AI. And Jakob and I met a few months ago in a summer school in the University of Copenhagen, where we actually worked jointly on some small projects related to the topics of what Jakob will talk about today. Jakob is doing his PhD studies broadly within medical image analysis, and with a particular focus on self-supervised learning techniques. With that, I would just like to welcome you, and the floor is yours.
Thank you. Okay, let's get the sharing sorted first. Okay. Could somebody just confirm that you're seeing the right screen? Yes, okay, perfect. So, first of all, Alexis, thanks a lot for the invitation. It's a great opportunity to be here, and also very nice to be in person in Lund. So I had the opportunity to see some of the city, and it was very nice this afternoon. So the topic of this talk is titled Medical Image Analysis with Limited Labels. And I think this is something that could be interesting to more people than just in medical image analysis, hopefully, because whoever has been doing some work with interdisciplinary teams basically know the hassle. If you want to do some new task, usually there's new labels required. So the way I would like to structure this is I want to talk a little bit about fetal ultrasound research projects that are going on in Copenhagen and in Lyngby, which my PhD is also part of, because I think these are nice case studies for the collaboration between medical researchers and machine learning researchers. And I want to use two case studies that I haven't co-authored, but I think are cool examples, and look at them a little bit from the point of view of what's the collaboration like, and then also what is the role and the cost of labeling in these projects. And then for the main part, we'll be motivated a little bit by, okay, how can we reduce this labeling effort, right? And we'll look into two projects that are very recent research. One is on the self-supervised learning for 3D brain MRI segmentation. And the second one is on unsupervised, completely label-free during training, detection of fetal brain anomalies. Okay, so first of all, here's some of the faces that I usually work with. It's a great team consisting of medical, clinical, and computer science researchers. Specifically, I would like to highlight my supervisors, Mads Nielsen, my main supervisor from University of Copenhagen, and also Åsa Bergen from Denmark's Technical University. This is in Lyngby, just outside of Copenhagen, so technically not Copenhagen. And Martin Tolsgaard, who's from the medical department from University of Copenhagen. Before we can start talking about fetal ultrasound and screenings and so forth, let's just get a little bit of an overview. So just need to get the Zoom. Okay, so the interesting thing about the fetal ultrasound screenings is that almost everyone knows something about them. Either you have experienced them firsthand or secondhand, or you've at least seen it on TV sometime. So you're going through them when you're pregnant almost everywhere in the world. And you typically have at least two screenings. So one in the first trimester. That's basically there to confirm the pregnancy, see if maybe there's multiple pregnancies, twins, and so forth. You can already do some basic checks. But then to really monitor how the child is developing, you do the mid-trimester screening in the middle, second trimester, to also be able to tell if the organs that are more developed look right. And then in the third trimester, these are typically optional screenings, and you do that to monitor the growth of the fetus. So if you have some reason to believe there might be a risk, for example, of growth restriction, then it's really important to be able to monitor. Okay, so if we talk about these challenges in detecting stuff on fetal ultrasound images, the good news is that most things can be detected with ultrasound. It's a pretty cheap and easy method to do so. However, the detection rates in reality, they vary quite a bit. So that's depending on skills and expertise. If it's an expert or somebody who's also doing a lot of other things, not just fetal ultrasound screenings. It depends on the equipment and also on patient factors, like if there are, for example, twins, or you have a patient with a high BMI, it can be more difficult to get the right images. All of these things are really related to image quality, and is always important, the image quality, but I think in ultrasound is especially important. If you look at these images here, if you've never worked with ultrasound images before, you might say, that looks very blurry. But to me, these are high quality images, so they can get a lot worse. And one of the main goals during the screening is to get these planes, which are basically slices through the anatomy at some predefined points. So you want to be able to visualize these planes and then visualize the anatomy that you should see there. And that's how you interpret these images. It's quite difficult to get the right planes. I tried to do that. That's a very low quality image. I tried to find some standard planes here myself, and it was very hard, but it was doable. And especially because I tried this also with some AI assistants that was telling me when I was in the right place and what to do. So that was a project mainly led by the PhD students here, Maxi and Sarah, and their supervisors, of course. And what they did was they developed a model for the standard plane classification. Is the image in the standard plane or not? And you could imagine that's like a standard computer vision task. Simply build a classifier. But actually what's really important to a clinician is to also understand, why is it a standard plane, or why is it out of plane here? There are, of course, some clinical guidelines that determine how that should look like. And what they did here was they built a concept bottleneck model, which first extracts some segmentation masks. So you try to segment the anatomy that you're actually interested in. And there's some image level concepts as well. And on top of that, you're doing your classification. That way you get an interpretable result that is also explainable. So why is this a cool example for collaboration? I think it's clinically motivated. It's an important problem from the clinical point of view. You have the explainability and the guidelines included. And then you also have some clever solutions to the technical challenges. I won't have time to get into the technical part here. But there's also solutions from the method sides. And then in the end, you also have, of course, the expert annotations. And here, I think this is actually not a lot of images that they labeled, but imagine almost 4,000 images with a total of 14 different segmentation concepts, and then up to 27 image label concepts. This is a lot of work. And I think this is work very well spent. But if you think about what makes this project cool, I think it's because they solved the right task in the right way. And then the labeling is what comes after. So it would be cool if you can reduce that latter part as far as you can. Second work that I wanted to highlight is on preterm birth prediction by Kivas is the first author, and then also Emilia from the clinical side. And what they looked here is preterm birth prediction. What you do there is you measure the length of the birth canal usually. And that's all you do in the clinic. That's kind of the gold standard. You capture an image of the birth canal and then you measure the length. And that works relatively well, but you can imagine that you can improve quite a bit on this. And it's very important because this is one of the key factors actually causing neonatal mortality. What you can do is also take in other information like the segmentation, some textual information from the area around the birth canal, and then also some spatial information about how much are things actually spaced apart, how much are we zoomed in. And here they used two pre-trained models. So one thing is they used the same segmentation network that I showed before. Then they also used a classifier that was also already pre-trained and just extended it with these other modalities. So it's a clever use of using these pre-trained vision models. And I think it's kind of inspiring to think about how could we bring in more of these? So how could we make more use of the pre-trained models? And of course, if you're in computer vision, maybe you think like, okay, this is like a old news. Like we have models like Dino V2, which are incredibly powerful foundational models that have amazing out-of-the-box features. Like here in this example, that are very semantic, and why don't you just use that? So doesn't that solve your problems? And it is useful for many, many applications, don't get me wrong, but it's also important to realize that the training data of these models are completely different. And if they, by accident, also contain some medical data, it's not in the detail that we actually require. Then also, just because we're working with images doesn't mean they're all the same. So we actually have also different kinds of challenges. One thing is, especially in the ultrasound use case, as I talked about before, the concept of image quality. So it's, of course, also important in natural images, but it's just to a different degree here. It's just the image quality is very deciding here, deciding factor. And then you also have things like 3D images in MRI, for example. So here you all of a sudden you don't have an image, you have a volume, and the memory footprint is completely different. And then lastly, you also might have multimodal data. We just talked about tabular data before. You may have some patient information. You may have different images, ultrasound and MR. And then maybe you have some lab results, and then also just some doctor notes that are completely free-form text. And these also sometimes need to go together. So I would then go into the first work, basically, that is trying to make a step into this direction, that we have better foundational models to solve some of the tasks. And this one will be going away from the fetal ultrasound into adult brains, actually, into MR imaging. But before we go there, I would just ask if there's any clarification questions. So I would put some time in the end for discussion, but maybe there's already some questions to the first part. Okay. Right, I don't know if I can see the chat or anything, but I will just...
I follow the chat.
Okay, perfect. All right, then we'll get started. So this work, we called it AMAZE. So it's on augmented masked autoencoder pre-training for 3D native segmentation of public brain data. And what's here actually the problem? So the motivation is that if you've worked with segmentation models before, then you know that actually still in 2024, quite surprisingly, UNets still tend to be the best solution, at least in medical. And there's not really any pre-trained models available for MRI segmentation that are trained on large-scale data. So the current state of the art there, you could say, is the Swin unit R, and I will talk about it a little bit later. But there's definitely room for improvement. So our goal in this work was to develop such a 3D segmentation model for brain MRI data, and specifically focus on the question, how does it actually help to improve this low label domain? So we look at 20 labels, which is typically something that you may be able to get from a collaborator in a reasonable amount of time. Then how well can you improve in that regime? What is a suitable pre-training method, actually? And then also, what is a suitable architecture? So, of course, looking into UNet-style architectures, but there have been some modernizations, and what does work here?
I have a question.
Yes.
So 3D units here, what does the 3D refer to there? I mean, are we talking about some sort of video, or like what is the 3D part?
Okay. 3D part is, if I just go back a little bit. So if you look at this image, so you basically, you can visualize it like this. You have a volume of a brain, and that means you not only have like X and Y, but also Z.
Right.
So, and you can solve this similar to this visualization here by like going through slice by slice and just saying this is basically like 2.5D. You can just process multiple 2D images, or you take the whole thing, and that means you do also 3D convolution and everything like that. Thank you, guys. That's a good question, I think. Yes. Okay. And for the pre-training methods, I will give a little bit of a background here, because not everyone will be familiar, I think, although it might be very familiar for people coming from computer vision domains. But there's this quite a nice method, which is called masked autoencoder pre-training. So what you do there is quite simple. So you take an image and you mask quite a high fraction of the image. So you can go up to 75%, dropping those patches, put them through your encoder. So this could be some vision transformer or something similar. Get your patch embeddings, only of the ones that you actually have. And then you put these into a decoder to reconstruct the target without the mask. You could imagine that this is really nice for learning actual pixel level information, like where are things, where are they exactly? But it's also quite challenging. But it turns out that this is quite a nice way of learning representations, and you have quite a low number of hyperparameters that you need to tune in order to get this to work. And if this is not a new idea, although it has been really popularized 2022 by this meta paper that have been just showing how to do it and how to scale it. But the idea is quite old, using these denoising autoencoders, where the basic idea is you have your signal, you put some noise on the signal, and masking here is one type of noise. Then you try to map your corrupted signal back to the original data manifold, basically. That way you learn the data manifold. Okay, so the problem was we couldn't just get started on this because there was no data set. So we looked at our state-of-the-art competitor, basically, and the Swin unit R here was trained on 5,000 CT images. May seem like a low number, but again, remember, this is 3D volumes, so every image actually contains quite a lot more information. And it was covering all kinds of anatomy, not just the brain anatomy that we were interested in. But on the other hand, there were also all of these brain MRI challenge data sets that were publicly available. So we thought, okay, maybe there's a way to just pre-process them in the same way and build the data set ourselves. So we collected this data set that we called Brains 45K, because it's almost 45,000 volumes. And it's from all of these different sources that have just been pre-processed in the same way. So basically spacing them in the same way and normalizing them to the same interval. And now we had like it's a mix, but it's also what you expect in the real world to have an heterogeneous data set covering quite a lot of different sequences and also different lesions. Then the next question is, we have our data set, how do we pre-train? So again, looking at the state of the art here, Sweeney et al. Was using masked reconstruction, but also some contrastive learning loss and rotation loss. So we tried that on the data set with basically the hyperparameters that they provided. We figured out that at least the latter two didn't really work out for us. There was the rotation loss, which was very, very easy to learn. So after just a few epochs, our models basically knew how to rotate things. They knew which way is up, so don't get any signal out of that task anymore. And then the second one with the contrastive loss, we just couldn't get it to work. Because if you're doing 3D models, then you're forced to do quite low batch sizes because of the memory footprint is just exploding. So yeah, and it's quite well established in the contrastive learning literature that you need very high batch sizes actually, so that things don't collapse. And that's what we saw here. I don't know if this is possible to make it work. We at least couldn't see the benefit because it's also doubling the memory consumption. Okay, so what we were asking then was, can we get some extra benefits out of this reconstruction task? And one thing that we were interested in is the robustness aspect, of course. So typically this is done through augmentations. You want to be invariant to some intensity shifts. Like if you go to a different scanner, to a different hospital, things that might look just a little bit different kind of noise, different kind of intensity, and maybe there's some artifacts even. But then you also want to be covariate to the spatial changes. So if your brains are rotated all of a sudden in a different way, they're differently scaled, or you have some elastic formations on the anatomy. You also want to be able to detect this and still work if you have a bit changed anatomy there. So as I said, this is typically done through augmentations. It's also the way we approach the problem, but in a slightly different way. By using the augmentations also during the pre-training. This is something that's usually not done because one assumption might be, if I have a pre-training data set that is large enough, maybe I don't need the augmentations. The augmentations are there to make my data set artificially larger. But we found that it's actually, in this case, still giving quite a nice improvement. The way we do this is we apply to our volume during pre-training both spatial and intensity-based augmentations. And we do the masking part, where we mask about 60%. We have any encoder backbone. So this could be like a unit encoder, for example. And then we have also a lightweight decoder. Now the reconstruction that we get here, of course, should unmask the image, but it should also reconstruct the spatial augmentations that we have applied. But it should not, in this example here, it should not reconstruct the intensity-based augmentations. So in other words, we have applied some intensities to the images, to the volumes, that we then reverse through the masked autoencoder. So we have some denoising step in there on top of the masking. Yes.
What is lightweight decoding?
In this case, it's like a unit decoder, but with only one layer per block. So basically also convolutional decoder, but using a lot less parameters than if you use a conventional unit, for example.
And this works better than having the whole decoder?
Yes, so one reason to do it is just to save computation. But actually it turns out that the representations are also getting a little bit better. So one way to think about it is you shift more of the expressivity into the encoder. And then even if you don't have that much in the decoder, you're still able to reconstruct. So I'll show some ablation on this as well. Yes, and then you do your fine tuning, and we just transfer the weights from the encoder decoder we disregard, and then we keep the unit. We get a normal unit decoder, and we also use skip connections. So this is basically our normal unit, but the encoder weights are transferred. And we don't use skip connections in the pre-training. And then we do our segmentation task. Yes, so this is an example of a reconstruction. So you can see on the left-hand side the input volume, and in the middle is the masked input. And then on the right-hand side, you can see the reconstruction. And what you can see, of course, is that the left-hand side looks a lot more grainy. There's a lot more of this like very high frequency detail. But if you look at the anatomy, you can see that actually there's quite a lot of the anatomy quite faithfully reproduced. And this kind of blurry image that you get is very typical if you do like a per pixel mean squared error loss, because you then kind of you incentivize the network to just guess kind of in the middle of whatever you have in terms of noise, so you don't have any kind of perceptual loss or something like that.
So the original there on the left, does that include these intensity augmentations on it? Like, or is it a raw recording there? Or have you done some augmentations or something on the original to the left?
That's a good question. I'm actually not sure in this visualization.
But that's at least what you do. I mean, that's
exactly
in your
yes.
pipeline you do that. You do augment it. And then
yes. So I'm not sure because I don't know where we pull it out.
Right.
But I think this is the input after augmentation also. Yeah, let's look at some results. So I think it's not so interesting to talk about the raw numbers that much, but just so I can go through what we did. So these are all from scratch trained models, so without the pre-training. And what we tried was 3 data sets. So RETS21 is a tumor segmentation data set. Then IELTS22 is stroke lesions. And WMH is white matter hyperintensities. And we did cross-validation on WMH and using the test set. Interesting thing here is that the test set is actually out of domain. So that is using, I think, different hospital, I think even different country, I'm not sure there. But these are basically out of domain images. And in terms of models, what we tried was the base unit, then an XL variant with twice the number of filters, the unit, and MedNext, which is a recently proposed modernization of the unit, which is more, maybe familiar with the ConfNext model. It's more transformer inspired from the architecture. Then also Swin unit R is our baseline. Yeah, and this is die score, and you can see that actually it's quite challenging. So that these are not very high die score points, but it's also just using the 20 labels.
What does the underline mean? Is it particularly 56.15? Is it underlined for some reason?
It only makes sense once I show the whole table. So I just cut this up a little bit for the sake of the presentation. But yes, so what you see here are the results after pre-training. And so bold is the best result, and then underlined is the second best. And what you can see in green and red is basically the change from going with no pre-training to pre-trained model. You can see that on average, we get like a 1 to 2 die score point improvement. In most cases, sometimes higher. And then we also have the MedNext M where we actually are a bit lower, surprisingly. But that one also had very good baseline performance here. So yeah, you get on average a 1 to 2 die score point increase across the board. And of course, this is not life-changing in terms of what you can do now with less labels, but I think a step in the right direction. And we were quite happy that we were able to also beat the baseline with a much more efficient method. And we looked into some of the choices of the pre-training. And one of them was the decoder. And yes, we talked about this before. So using a lightweight decoder, it gives you about 25% speed up. And then also it gives you quite a bump in downstream performance. So you can get up to like 2 die-score points improvement in this example with the UNet. And also the other interesting thing was the augmentation scheme. So does it make sense to do this trick with your augmentations? Yes, it also gives you a little bit of an improvement. Also on average, in the ablation experiments we ran, was about 1 die-score point improvement. Okay, so what are the takeaways of this one? So we built the first actually large-scale data pre-trained model that is specifically designed for segmentation for this 3D brain MRI data. And we showed that the pre-training benefits downstream performance and across the tasks we look at in this few-shot setting that we investigated. And also that the pre-training improved out-of-domain generalization in this WMH dataset. So that was the first research paper, and I have a shorter third part as well. But are there any questions up to now?
Yeah, one question is, like, did you have a difficult time deciding these augmentations themselves? I mean, you had a bunch of augmentations, some of which it should learn to revert, and some of which it should sort of not revert in some sense.
Yes.
So, like, was there a lot of thinking there, or was it quite straightforward, that part of finding these augmentations to use?
Yeah, I think you could definitely fine-tune that more, which augmentations to use. But our approach was, so we had a number of typically used augmentations, and we just sorted them into which change kind of the spatial data and which just change intensities, including the ones that actually do some artifacts. We might have some new like some ghosting or something like that in the images. We also regarded them as intensity based.
Right.
Yeah. But there might be, there's always, like, with more augmentation, you can, I guess, get more performance in the end. Yeah.
I also have a question. Can you hear me?
Yes.
Yeah.
I was wondering, I was under the impression that somehow this masked image modeling or masked autoencoder stuff works better for vision transformers. But this was all convolutional, what you showed it.
Yes.
Maybe I'm simply mistaken, or have you tried using a vision transformer for something? Or why do you use convnets?
Yeah, so we used convnets because in the medical literature, the segmentation networks that work best are typically the UNet-style convnets. So it's like there's just been some recent papers again benchmarking like NN-UNet, which is like a well-tuned and self-configuring UNet. And it's just for these medical segmentation tasks, seems to be the ones that still achieves best performance. So I think you're definitely right, and using a vid transformer or something like that has a lot of benefits, especially when you mask. You can actually drop tokens. You can do that with convolution, but it's not so simple. But yeah, we were motivated to try this with the convolutional architectures because we knew that because of the inductive bias and so forth, they work quite well on the segmentation tasks.
Right, thanks.
Okay.
I have a bit of a question. I'm not really into this, but so more a biological point of view. When you're removing 75% of the pixels from the image,
yes,
and see if you can recreate it to do, and I guess this is for these 3 scans during the pregnancy. But so the goal is to have an image and see if you can actually get the same results on something where you only have 25% of the image. But isn't it so that when you have these scans, it's not like you're missing 75% of the image. All of it is blurred.
Yes.
And how does this then help in getting the right results from these blurred images when you're actually training the model of when you're trying to get something more clear on an image that is not 25% of a perfect image, but is 100% of something that is really blurred?
Yes. So we're not necessarily using this masked image modeling thing to actually be able to, in the end, process corrupted images. It is something that you may be better at, but the main reason why we do it is actually because if you just have a lot of images and you don't know what is contained in the images, that's one way to learn without adding any additional supervision. For example, labels of this image contains a cat and here's the head of the cat. But you just remove some information that you already have and you know what you removed. So you can give some training signal of what was there where I removed the patch. And it's just a way of basically learning from the data without requiring any new information except what you already have. So, yeah.
And to continue that, just so basically, this step of the mask encoding, when you have done that training step, then you can use that AI model, so to say, to kind of tune it to different other tasks. One task could be the task that you talked about, the blurring some actual images. But in your case, the task was segmentation.
Exactly. Yeah.
Okay.
Yes.
So it's like a pre-step. It's not the end goal, really.
No.
Yeah.
Okay.
Yeah. So it's just one way of doing this self-supervised learning, basically. Okay. So the last project, I didn't say this before, but both of these are actually going to present it next week at Mikai workshops. This one at the Asmus workshop. So this one was, again, coming back to fetal ultrasound. And this is an example for unsupervised detection of some brain anomalies in the fetal ultrasound setting using the diffusion models. And so basically, can we also do something without any labels? So yes, I think I should give a quick introduction into what fetal brain anomalies are or what they look like. So in general, it's quite rare to have any congenital malformations during pregnancy, but if they happen, one of the more frequent ones are the brain malformations. So it's one of the things that you try to detect in this mid-trimester scan, and it's a very challenging problem because we talked before about these brain planes, and one problem here is that you may have a healthy brain and you just look at it from a different angle than you're used to, and you get a completely different picture. So you have very high intra-class variability. And you can, on the other hand, have very low intra-class variability. So it can be that there is a cyst in this brain, but it's just a very small part. It's just a few pixels, and the rest looks just like you're used to. So this is a challenging problem just from the computer vision point of view. And then this also being a anomaly detection task, you have this typical long-tail distribution of events. So a lot of things, they are already rare, but they get just more rarer, and you have a lot of them. So it's difficult to pick up all of these classes. And the motivation here, again, that we got from clinicians and talking to sonographers that are doing these scans is they're not actually working like a computer vision classifier that have just learned every class. They have learned how the normal healthy brain looks like, and then they detect if something looks off, basically. So this is more like an unsupervised learning approach. And if we look at some previous works in this area, they have been all supervised. So they were requiring labels on the image level, so classification labels, or even anatomy labels that were going down to bounding box level. And therefore, you're also restricted to a finite set. So you can only look at the most frequent ones that you actually have the training data for to have these sort of classes. And they were also going to quite a thorough data cleaning pipeline and extracting a lot of the same images from the same videos, a lot of images from the same videos from the same patients. So these were some of the things that we wanted to look at and see, can we go with an unsupervised approach, and can we also do less data cleaning and have something that's a bit more representative of the data that we're seeing. And so I think this part will probably make more sense to the people that know how a diffusion model works, but I will try to just explain it briefly in 2 sentences. The basic idea is you have a model that you take your images and you noise the images. You apply some noise, for example, Gaussian noise, and you learn how to reverse noise at a single step. It's kind of a related idea to what we've seen before. The important part is here that you're really only doing this, going from more noise to a little bit less noise. And in the end, what you can do with these is going from pure noise, you can recreate images that look a little bit like, or that look like your training data distribution. You can use different types of noise. So usually you use the Gaussian noise, but in other anomaly detection applications, people have experimented with these pyramid and simplex noises. The idea behind this is that Gaussian noise basically gives you equal uniform distribution on the noise that you apply, on the frequencies that you change, while the images themselves are more dominated by the low frequencies. So you have more low frequencies in the image. So what happens if you just apply Gaussian noise is you remove more of the high frequency detail. The low frequency detail stays the same or stays less affected. So people have been successful using the pyramid or simplex noise that has more low frequency components. Yeah, and here you can see some examples of the initial image and then noise to 150 noise steps and then recreated. And it's a little bit hard to see, at least here on the projector, but the Gaussian noise at the highest fidelity, but that's also not the use case for pyramid and simplex. So we found it very interesting to also look at these different noise types because they haven't been looked at so far that much in fetal ultrasound. And now, how do you use it? So we have a model that can do this, can produce images and can reconstruct images. How do you use that for anomaly detection? So during inference, we're done with training. We take some input, and that may or may not contain some anomaly. So in this case, we have this anomalous region here, which is a cyst, and it has been circled by the clinician we were working with. This is not something that is actual input to the model. The model doesn't see the red circle here. And we corrupt the image, and we do not only do that once, but we actually do it to different degrees. So we corrupt it a little bit, and a little bit more, and a lot. And so we have these 3 images that are corrupted to different degrees. And that's a hyperparameter, of course. You can do that however you would like. What we also do is we use a segmentation model to mask out the fetal head so that we only apply this noise to the head region. It's called in-painting. And then we reconstruct, so we basically in-paint the area within the head. So the model is trying to reconstruct the image. The idea here is that because the model has only seen how healthy brains look like, it has some noisy area and will try to reconstruct also a healthy brain that looks kind of like what it sees there. So what you can do then is you can take the average of all of these in-paintings, and you can compare that to your original input. You can, for example, do this difference map, where you can then see which areas are actually the most different. And then this is like a nice example where you can actually see highly localized change. But this gives you some interpretability of why something is an anomaly. Then what you do to actually do the anomaly detection is you can just apply some metric on the reconstructed image and the original. You can use, for example, we used structural similarity loss, which is a pretty simple method, but we found worked best in this case. And then there's some more examples here. So again, our clinician had circled these areas, and we can look at some examples like the hydrocephalus example here on the left. You can see that what the clinician has circled was this this ventricle down there, and it's something that was picked up from the model, but actually a lot of other areas were also changed. But it turns out these are also largely where other ventricles are, so it's also actually changing in other places, maybe not just the one that you would that would pop out immediately. But it gives you, in many of the examples we looked at, it gives you some degree of explainability, but it's not enough fidelity, of course, to do like a segmentation map out of it.
A question here.
Yes.
Like, I guess it depends on application and stuff, but would you say that typically, like, is precision or recall most important? Like, you had an example there where it sees a lot of other red regions, not just the one that didn't think it. So I guess the recall is quite high, but the precision is maybe not so high sometimes. So,
yeah.
Like, do you get my question roughly?
Yeah. So we looked at rock curves primarily in this one, and there's like, I think, a different question: how do you calibrate those? And I think that's very much in the clinical side almost then. But yeah, we saw some examples which had more precision or more recall. It was also kind of dependent on the kind of anomaly. And I'll get into that a little bit in the next one. Because there are some differences in this, right? So you have some that basically affect the whole brain. Then you have some that are very localized changes, such as the one that I showed in the very beginning. And actually with this method, we got better performance with the localized regions. Yes, so let's look at some of the ROC AUC values. So we also had a supervised baseline, which was just a ResNet 18 that was trained on normal and abnormal images as a binary classifier. And we expected this to be actually way better than what we could achieve with the unsupervised approach in many of the cases, because we had some of the training examples, so did heavy augmentations and stuff like that on the ResNet. But it turns out that there are a lot of cases where you can get quite close to the supervised model. And then there's these examples of anomalies that are very locally manifested, like the cerebral cyst or ventriculomegaly, which we also looked at before. And these were quite well detected by the unsupervised methods. Yeah, and we looked at the different noise types, and we found out that actually the Gaussian noise overall seems to be performing the best. Yeah, I don't want to go too deep into the ablation studies, but we looked at the different components of the method, and so we tried first with perceptual losses that are using other neural networks to extract some embeddings, but we couldn't really make that work even with networks that were trained on fetal ultrasound data. But we had more success just using structural similarity index, and then also adding the in-painting step and adding the averaging in the end. So having more noise steps basically to different degrees. Okay. So the takeaways of this research project, I think, for us were that the task was quite challenging, actually, on the data set we had here, also indicated by the supervised baseline performance. And with the unsupervised method, we could match or exceed the baseline in some cases for these locally manifested anomalies. So it was working quite well. But the non-local anomalies, especially the ones that affect the whole brain, were quite difficult to detect with this one. And the Gaussian noise was generally working best, but that's also interesting. It seems to be something about the noise of the images themselves. because in brain MRI, for example, it has been shown that the simplex noise seems to be quite well performing. Yes, and I also want to mention with this work that there was a master thesis by Markus Olsen that I could supervise. So I think that was also very nice work there. That's all I have for the presentation. So thanks for your attention. I look forward to some discussion.
Yeah, great. Really great presentation. Thanks a lot. So yeah, I just want to repeat what you said. Like, let's discuss, and if you have any questions, go ahead. Both from online, of course, and if there's questions in the room.
I can start with this. You looked at noising the images and then reconstructing them in this final paper. Could you imagine using in-painting, or was that what you mentioned in-painting? Or was that instead of the noise?
You mean in-painting a different image?
Well, no, taking the image and then taking away a part of it and then letting the diffusion model reconstruct that part of it.
Yes, so that is
like you could do random patches or something.
Yes, so you mean like going even more extreme with the deterioration of that part,
exactly.
Basically. So yeah, so the problem then is, I mean, I'm sure that there are some ways in which you can make it work. If you take out the whole brain, of course, you lose all of the information of what's already there. But maybe if there's some areas that you're specifically interested in, then I could imagine that it could make sense to like just apply more noise to those areas and paint like specific structures, maybe. Yeah.
I mean, if that patch will accidentally be over some anomaly, it will probably be reconstructed without the anomaly or something.
Yes.
And then you would have a big difference to the input image.
Yes. So basically the assumption here was that even if the anomaly is still a little bit visible through the noise, that because you're going back to your original data distribution, you would remove that also as part, basically see that as part of the noise that is there. Yeah, so you would remove that because that's not something that you would see in the training data distribution.
Yeah, so I was thinking about these Gaussian and the other types of, I don't remember, pyramid things that you mentioned. So question one is, in general, is the approach there, is it slow? Like, I mean, using diffusion and stuff, I mean, generally, like, what's the compute time for doing a forward pass of? And maybe you don't remember the exact number, but order of magnitude, is it a fast process in general?
It's pretty fast. So in this one, I didn't actually run the experiments myself, so I couldn't tell you exact numbers, but it is pretty fast on the inference. But of course, the more you do, like adding multiple inferencing steps, like multiple noise levels and so forth, you have a trade-off there.
But it's not like really a bottleneck, the compute time.
No, I wouldn't say so. No.
And follow up to that, and given that it's quite fast, I mean, did you try to do some sort of ensemble of the different types of Gaussian and pyramid and stuff? Like, could that be the best?
I'm not sure actually if we tried that. I think it was something that was discussed at some point. So I don't think we tried it in the end. But yeah, it's definitely something that comes to mind. Like, why not combine the strengths of those? Yeah.
You know roughly how many steps in the diffusion process you use?
So there's like 150 noise steps.
Okay.
Max, I think. I don't know if there was some cases with the Gaussian noise where maybe we used a bit more. So, yeah, these are basically the noise steps. So up to 250.
Check if I know if somebody has chatted or something. Otherwise, I have more questions.
Yeah, please come with questions in the chat or just unmute.
So I have another question, and it's the masked autoencoding is for the 3D case of the thing that we talked about. Like, do you do it the same in the time, I mean, the same pixel, or if you will, in the time dimension all the time? Or can that switch between time steps if you get the question? Like generally, if you have a video,
Yes.
is it happening in the same pixel throughout time steps there?
Okay. So there is not actually a video. So
No, I get that.
Yes.
But there's still a temporal dimension, right?
No.
Okay.
Not really. So
Free spatial dimension.
yes.
Okay.
So it's just like a spatial volume, basically.
But the maskings looked like they were sort of three-dimensional boxes.
Yes. So we used like these patches,
Yeah.
like you would do when you do it with a transformer model, even though we didn't do actual tokenization of the image, but we cut out these square patches, basically. That was also something where we experimented a little bit with how large should these patches be. And we figured out that if they are too small and then the masking ratio is too low, and it's just a little bit too easy. So these kinds of things are quite difficult to do cross-validation on because you train for a long time in the pre-training. This was something that we ended up with that seemed to work quite well. There's actually an interesting question in there because you do the 75% with the natural images in 2D. So what does it actually mean to mask in 3D? Is it more difficult or is it easier because you can look at the patches from different directions? So I think that's also interesting direction.
So have you, or do you have plans on trying some of the ideas that you're doing here for the medical domain, like trying them out for a more standard imagery, natural imagery, or are you still going to mostly focus on the medical setups, you think, in the future or coming years?
Yeah, mostly focusing on the medical setups because we're really motivated by this fact that the big models, big vision models, for example, don't really seem to work on the medical data. And it makes sense. I personally also think it's not good to think too much about all of these different images just as images, because they are like really different modalities. So of course, there's always going to be some like transfer learning from one to the next. But then the question really becomes like, how much do you just learn like basic features by pre-training on other natural images? How much do you actually, I think what would be nice would be to have a better representation of actual anatomy and also the imaging modality.
So maybe if there are no questions there, I guess maybe we should conclude the session and I'll just come up from there. Cool. So let's thank the speaker again. Very interesting. I haven't worked myself so much in the medical domain, so that's very interesting to hear about the challenges in that area. So thank you again, and next time will be in 2 weeks when we have Matthias Obst at University of Gothenburg, who will be presenting, and he will also be actually in place in the Gothenburg office. So, see you online or in Gothenburg in 2 weeks. Thank you.